In this section of Bio 212, we've been talking about membranes and membrane function, and highlighted that one function of membranes is that they allow for compartmentalization of cells. This compartmentalization can be as simple as forming a plasma membrane that separates the inside of a cell from the environment outside. But in this microlecture, we will focus on membrane-bound compartments within cells, called organelles. Some of this material will be review for some of you, but even if it is, be thinking carefully about integrating what you have learned about protein structure and function with your understanding of membrane function to understand how organelles work. There is a diversity of membrane-bound compartments in different cell types. The presence of a membrane allows regulation of what does and does not cross, and allows each compartment to have a different mix of molecules within it. This mix of molecules, and especially the mix of different proteins, gives each compartment a specific function. Prokaryotic cells, cells without nuclei, have an incredible diversity of structures that we could discuss. But in this microlecture, we're going to focus on just a few organelles found in eukaryotic cells, including the nucleus, mitochondria, chloroplasts, and components of the endomembrane system. Note that cells are packed with other complex structures that we'll discuss during the course, but that true organelles are surrounded by a membrane, and we'll focus on these membrane-bound organelles today. All eukaryotic organisms are comprised of cells with a nucleus. The nucleus is surrounded by two lipid bilayers that form the nuclear envelope, and contains the vast majority of the cell's DNA, and is the location at which genes encoded within DNA are transcribed to make RNA. We'll discuss transcription and the functions of RNA later in the term. Molecules moving between the nucleus and cytoplasm cross the nuclear envelope through nuclear pores. Like all membranes in the cell, only some molecules can cross the nuclear envelope, allowing the nucleus and the cytoplasm to have different molecules present within them. We just used the term cytoplasm, which refers to all of the space inside the cell but outside the nucleus. Another term you will encounter when discussing organelles is the cytosol, which is the space in the cytoplasm that is not within any organelle. Moving on to other organelles, the mitochondria are the location at which the majority of ATP synthesis takes place in eukaryotic cells. There are hundreds or thousands of mitochondria within each cell, and each contains a complex set of enzymes that catalyze chemical reactions that ultimately use oxygen as part of a process that makes ATP as an energy source for the cell. We'll discuss this process further next week. Mitochondria contain two membranes in their own DNA, both remnants from their origins as ATP-producing prokaryotes that were taken up by a primitive eukaryotic cell and now are permanent organelles within eukaryotes. Chloroplasts are the organelles in which photosynthesis occurs in plants and other photosynthetic eukaryotes. Chloroplasts also were once free-living prokaryotes that evolved into permanent eukaryotic organelles. The remaining organelles we will discuss in this microlecture are all connected as part of the endomembrane system. This system of organelles includes the endoplasmic reticulum, Golgi apparatus, lysosomes, exocytic vesicles, endocytic vesicles, and other vesicles that move between organelles. The organelles of the endomembrane system work together to modify and transport proteins, lipids, and carbohydrates targeted to the cell surface, to bring some molecules into the cell, and to degrade macromolecules, among other functions. Think carefully about these multiple functions as we introduce the organelles of the endomembrane system. The endoplasmic reticulum, or ER, is where most macromolecules that will end up in the plasma membrane or outside the cell begin their journey to the cell surface. The rough endoplasmic reticulum appears rough because the membrane is coated with ribosomes, which synthesize proteins that are immediately transported into the ER. The smooth ER is where specific phospholipids are synthesized and inserted into the membrane. These phospholipids eventually end up being transported throughout the cell to form the membranes of all the organelles. Small membrane-bound vesicles leave the ER and carry their contents from the ER to the Golgi apparatus. In the Golgi, proteins and lipids brought from the ER are modified, often by having chains of carbohydrates linked to them. Vesicles also leave the Golgi, carrying proteins and lipids and their attached carbohydrates to other locations within the cell. Some vesicles move from the Golgi to lysosomes, organelles that are packed with enzymes that digest or hydrolyze proteins, carbohydrates, lipids, and nucleic acids. The inside of the lysosome is acidic, so these proteins together are called acid hydrolases. This leads us to two thought questions. Number one is, what would get pumped across a lysosome's membrane to make the inside of the lysosome acidic? And number two is, why would a cell want its lysosome to be acidic, while the rest of the inside of the cell is of a neutral pH? Think about these questions for a minute, then check the slide at the end of the microlecture for some answers. As we continue through the endomembrane system, consider that we have thus far described the movement of proteins and lipids from the ER to the Golgi and then to the lysosomes. But earlier, we said that a major function of the endomembrane system is getting macromolecules to the cell surface. Some vesicles, called exocytic or secretory vesicles, depart the Golgi for the cell surface. When exocytic vesicles reach the cell surface, the vesicle membrane fuses with the plasma membrane, resulting in the release of vesicle contents into the space outside the cell. Molecules that are secreted outside the cell using exocytosis include some hormones. Insulin is one of these secreted proteins that you have likely heard of, and proteins that link cells together and allow them to communicate. Our cells would not stick together if it weren't for the regulated exocytosis of specific proteins, lipids, and carbohydrates that cause cells to attach to each other. The fusion of an exocytic vesicle with the plasma membrane also results in the integration of molecules from the vesicle membrane into the plasma membrane. Molecules that end up within the plasma membrane include specific phospholipids and integral membrane proteins, including many of the transmembrane channel and pump proteins we are talking about in lecture this week. The process of secreting material from the cell using exocytic vesicles is called exocytosis. Finally, endocytic vesicles bring proteins and lipids from the plasma membrane and extracellular space into the cell, where they may be transported to lysosomes or to the Golgi. You saw a specialized endocytic vesicle, called a phagosome, capture and internalize a whole bacterium into a neutrophil in the video we watched on the first day of class. The endomembrane system of organelles, the ER, Golgi, lysosomes, and vesicles, work together to coordinate the movement of important macromolecules between the inside of the cell and the cell surface. Can you think of any proteins that you've already read about for Bio 212 that are likely dependent on the endomembrane system to perform their function? What role does the endomembrane system play in aiding these proteins with their cellular function? We'll think about these questions and others as we continue to address the interactions between proteins, membranes, and whole cells. You are now ready to put together what you know about protein structure, membrane function, and organelles to think integratively about protein localization in eukaryotic cells. We will apply these concepts to a specific protein required for normal function of several cell types, including lung cells, in class this week. Answers to the lysosome questions are on the next slide. Please be certain to get in touch with your instructors if you have any questions about this microlecture or any aspect of the course.